Hello everyone children, good afternoon. So we are going to see an important practical now which is very much involved in biology. Whenever it comes to biology, we always talk about tiny organisms which can be magnified. There are few organisms which cannot be seen with our naked eye. In lower classes you could have learned what are microbes. These are tiny organisms which cannot be seen with our naked eye. So through what we can see? We can see using instrument called as microscope. Microscope is actually an instrument used in the field of life science, that is biology, used to magnify the tiny object which cannot be seen with our naked eye. This mostly we are using to visualize the specimens, biological specimens. I'll tell you how to hold the microscope. Before that, let me tell you the parts of a microscope. Fine. First, this is called as arm of your microscope. If you are right-handed, hold the microscope like this with the help of your arm. The lower area is called as the foot, which is generally horseshoe shaped. This is called as the foot. This one is called as your foot. With the help of this foot only, the entire weight of the microscope is held. So whenever you handle a microscope, first you hold the microscope with your help of your arm and hold it with your left hand where the base, that is the foot, resting on your palms. This is how you have to hold the microscope. Not like while one having two microscopes. One person have to carry only one microscope at a time. This is the way we have to carry the microscope. Now let me tell you the different parts of a microscopes. The first important part in microscope is actually the base I am talking about. So this is called as the basal foot, which holds the entire part of your entire weight of the microscope is bound on this foot. Then we have a mirror here. There are two types of mirror, children. One is concave mirror, second one is called plane mirror. For bright light we are going for plane mirror, and for dim light we go for concave mirror. Or for artificial light we go for plane mirror, for natural light we go for concave mirror. This will collect all the light radiation from different areas in the surrounding. Nowadays we come with LED and lot of artificial light sources are also there, but generally this is the way we have to have a mirror. Through the help of this mirror only we have to collect light from various sources and we have to project towards the microscope. Artificial lights are also is provided. Next we can see one important structure here. You can see here it's closing, it's opening up. I'll just show you a little bit closer. You can see here it's opening, so the light can enter in, it's closing. This acts like your front part of your eye, the pupil, which actually regulates the opening and closing of amount of light that have to enter into your body, that is to your eye. Similar way, here we have the microscope, children, where we have the diaphragm and here we have the condenser. So what it will do is, it will adjust the amount of light. If you want to decrease the light, just close it, the amount of light entering will be decreased. If you want to increase the amount of light, just open it up, amount of light has to be opened. Always in a microscope, diaphragm will be closed to prevent from dust entering in. If you are using a microscope, open it up to the full, adjust the amount of light required and you can go with your experiment. Now I am placing the mirror over here. Next we have an important thing that is called as stage. What is the role of the stage? Stage is the area where we will be keeping the slides which is provided for you. And they will be provided with something called stage clips. So the stage clips are 2 in number. The stage clips are used to hold the specimen slide that we are going to keep it over in the stage. Then this is called as the body of microscope, arm of a microscope, foot of a microscope. Okay, as I told you, this is the body of a compound microscope. This is the arm, this is the foot. In the body of a compound microscope, there is one piece which is above. This is called as eyepiece, which comes under 10 times the magnification with 10x, 15x magnification and 20x magnification. So below that we have something called nose. This is the body, where the top of the body we have something called as eyepiece. Next is we have something here, this is called as nose of a microscope. The nose of a microscope is having another lens This is called as objective. It is called as nose of a microscope, where we have the objective. Objective mostly comes with 5x, 10x, then it comes with 45x, then with 100x, that is the oil immersion. So this can be removed by anti-clockwise direction. If you want to put it, we have a groove, just put it over here and you can just tighten it in the clockwise direction so you can attach it. Fine. Next is you place your eyepiece over here. Now there are two types of adjustments you can see. You have to use both your hands for experimental purpose. You can see one which is moving the microscope very fast. So this adjustment is called as coarse adjustment. This one will not move the microscope much. This is called fine adjustment. Coarse adjustment screw, then fine adjustment screw. So eyepiece, body of a microscope, nose piece, objective, coarse adjustment to move the microscope up and down, fine objective to get a fine clear image, stage to keep the specimens, stage clips to hold the slide. Then we have diaphragm, which may allow the light molecules to enter the amount. It will determine the amount of light molecules that have to enter in. Then we have a mirror by which the amount of light can be adjusted. What is the first thing you have to do always when we work with a microscope? Means you have to adjust the amount of light and we have to get a very big bright circular field. After that you have to place your lens and always you have to start with 10x magnification. And you have to see with the help of your eyepiece from the top only, from this area only we have to see. Once you get a clear image, then you can just have a fine tune to get a clarified and contrast image. Then you can go for higher magnification. After when you go for higher magnification, don't use coarse adjustment, use only fine adjustment to observe the specimens. Very important, children, that I want to state here is handling microscope is an art. And of course, it's every biology student must know how to operate a microscope, how to clean a microscope and how to safeguard the microscope. So these are the various parts of a microscope. Summing up, magnification of a microscope is equal to magnification of objective and magnification of eyepiece. Eyepiece 10x, objective 10x means 10 into 10, 100 times the magnification. Eyepiece 10x, here it is 40x means 10 into 40, 400 times magnification. I am using oil immersion, or if I am using water, maybe think. If I am using here 20x, here I am using again 10x here, 200 times the magnification. So magnification is nothing but how much times the image is magnified means it is determined by the power of the eyepiece and power of the objective. Multiply these two powers, you will get the maximum magnification. So these are the various parts of a compound microscope, which is very much essential and it will come for your examination also. I have attached today what you have to do for your experimentation also, what you have to write. I have posted in the classroom, children, kindly do it. Any doubt, kindly post and I am there to support you. So when the school gets reopened, children, you have to work on the compound microscope. You have to adjust the light and you have to name the various parts, and I'll be giving you the specimen. You have to observe in low power of the microscope as well as on the high power of microscope. So thank you very much, children.